filtration, reabsorption, secretion, urine formation, fluid and electrolytic imbalance and balancing. So filtration. First thing is filtration. Filtration is about once a blood enter into the nephron of glomerulus, the small molecules like water, ions, glucose, ammonium, everything are filtered into glomerulus of Bowman's capsule and forming the filtrate. Then the filtrate are reabsorbed where at PCT, proximal convoluted tubule, or Hen's loop, and DCT, which reabsorb the essential substance like water, amino acid, filtered substance like water, amino acids, ions, and back into the blood from the filtrate. Then secretion, which means the secreted waste product and excess ions under filtrate, which is eventually become urine. Then a urine formation. The finally filtrate, now called urine, is collected by collecting duct and transported into the renal pelvis. Okay, collecting duct and transported into the renal pelvis. Now the fluid electrolytic balance. So the nephron plays a crucial role in maintaining a body fluid. Okay, fluid like electrolyte like ions, sodium, potassium, chloride, everything. So balancing the controlling and reabsorption of water and ions. Then the counter current. What is the counter current means? It is there is a vasa recta. See, there is multiple of collaterals and capillaries are there. Vasa recta and counter current multiple system. See, the counter current multiple system is a mechanism which is involving or occurring in loop of Henle and collecting duct. Both the thick and thin, ascending and descending loop of Henle and collecting duct. The loop of Henle is divided into descending limb and ascending limb. There, the descending and ascending limb further divided into thin and thick segment. Okay, the descending limb is permeable to water but not solute. Which means an ammonic acid like that, which is ascending limb is permeable to solute. So that not water. So loop of Henle established the concentration gradient in medulla of the kidney, so creating the high concentration of solute in interstitial fluid of renal and medulla. So there the thick ascending limb actively transported ions such as sodium, chloride, potassium for the tubular into fluid, from tubular fluid into interstitial fluid. Okay, the concentration. This create a high concentration fluid where the ammonium, sodium, water, ions. Okay, these create a very high concentration of solute in the interstitial fluid, which draws water out of descending limb and collecting duct, which is leading to concentration of urine. See here, the loop of Henle, 300 ml of in NaCl and water 1000 ml, 1200 ml, again 1000 ml, 800 ml, and thick filament containing NaCl, thin filament containing water. This is what happening in counter current multipolar system. So the collecting descending limb and collecting duct leading to concentration of urine. The counter current multiple is called as counter current because flow of fluid in the ascending and descending limb of loop or Henle, which is opposite in the direction. So this current flow essential for establishment and maintaining the concentration gradient in renal medulla. And this is about counter current. And now going to see about vasa recta. Vasa recta is a multiple or network of capillaries that runs parallel to the loop of Henle. Henle is present in Henle. So vasa recta plays a many important essential role in counter current multiplier system. So it providing blood supply to the renal medulla. So vasa recta in a counter current exchange the system, which means the blood flow in capillaries is in opposite direction to the fluid in Henle's loop. This counter current flow allows from the exchange of solute once in a water between vasa recta, which means multiples of capillaries, and interstitial fluid in renal medulla, which means a vasa recta exchange the solute and water between the network of capillaries and interstitial fluid in renal medulla.